Hello, everyone. Today I am honored to introduce the paper submitted to iMetaOmics. The title is Electroacupuncture Reshapes the Microbial Co-occurrence Networks Related to the Behavioral and Psychological Symptoms of Dementia in Alzheimer's Disease. Although the gut microbiota is thought to play a crucial role in the pathogenesis of Alzheimer's disease, AD, its association with the behavioral and psychological symptoms of dementia, BPSD, in AD remains largely unknown. BPSD belongs to the spectrum of neuropsychiatric syndromes, NPS, encompassing more than 12 complex symptoms divided into 4 major categories. Microbial keystone species and gut microbiota composition are highly variable during the pathological development of BPSD and AD. Age stratification reveals stage-specific gut microbial signatures in AD-related BPSD. In this study, we investigated the effects of electroacupuncture on the dynamics of core microbiota and keystone species in the microbial co-occurrence networks of AD-related BPSD, as well as their crosstalk with the predictive metabolic pathways. The pattern of microbial dysbiosis in AD-related BPSD was identified using AP/PS1 mice, with C57BL/6J mice serving as the control. Fecal samples were collected from each group at baseline and 3 weeks post-intervention. The V3-V4 regions of the 16S rRNA genes were amplified using the 338F and 806R primers. Our analyses demonstrated that Bifidobacterium pseudolongum, Lactobacillus hamsteri, and Parabacteroides distosones were significantly enriched, while Akkermansia mucinifila and Clostridium cochleatum were significantly depleted in both 6 and 9-month-old AP/PS1 mice. The specific microbial markers that distinguished 6-month-old AP/PS1 mice from age-matched WT mice included Lactobacillus vaginalis and Rhinococcus navis, while those that distinguished 9-month-old AP/PS1 mice from age-matched WT mice included Alistipes phenegoldii, D-sulfovibrio C21 or C20, Faecalibacterium prosnitzii, Alistipes massiliensis, and Clostridium salatum. Due to the marked variation in the abundance of Rhinococcus flavescens in both age groups of wild-type, WT, mice, the comparison between AD and WT mice exhibited an opposing trend, a higher abundance in 6-month-old AP/PS1 mice but a lower abundance in 9-month-old AP/PS1 mice when compared to age-matched WT mice. Keystone species were identified based on centrality scores and intra- and intermodular interactions within co-occurrence networks, as generated by Pearson correlation coefficients. In the microbial co-occurrence network of 6-month-old AP/PS1 mice, Mucosphaerum schedleri and Clostridium perfringens were negatively correlated and identified as keystone species, together with Subdiligranulum variable, a diabetes-related species. In the microbial co-occurrence network of 9-month-old AP/PS1 mice, Bifidobacterium pseudolongum, Lactobacillus hamsteri, and Butyrococcus pullucicorum, which have anti-inflammatory effects and produce short-chain fatty acids, and Clostridium salatum, a risk factor for type 2 diabetes, were identified as keystone species. In the co-occurrence network of 6 and 9-month-old AP/PS1 mice post-electroacupuncture, Rhinococcus navis, which is related to inflammatory bowel disease, IBD, and produces polysaccharides, was identified as a likely keystone species. Our findings support our hypothesis on the coregulation of glucose, L-arabinose, and gallate degradation with chlorophyll A biosynthesis in the gut microbiota of AD-related BPSD following electroacupuncture intervention. Our findings suggest that electroacupuncture can ameliorate AD-related BPSD by driving keystone species in the microbial co-occurrence network and by regulating the composition and functional metabolic pathways of core microbiota.